All right, so thank you everyone for coming to the last of our four-part Tug Hill Winter Wildlife webinar series. It's not feeling very wintry out there at the moment, so we're gonna go ahead and wrap it up with a bang. Yeah, our first three webinars were on Fisher, Martin, and Tug Hill birds, and our final topic is brook trout. And I'd just like to thank our two presenters for tonight, Chris Murphy and Paul Miller from Trout Power and Trout Unlimited. We're very happy to have them, and I think they're going to present some really interesting information tonight. A couple of logistical things. Um, the webinar is going to be recorded, and tomorrow or in the next couple of days you'll get an email with the recording. And those are also accessible along with the previous webinars on our website or our YouTube channel. If you have any questions throughout, which are highly encouraged, you can just put them in the chat, whether they're logistical or about the information that's being presented, and we will get to most of those at the end of the webinar. We're shooting for around an hour, 45 minutes to an hour 15 max. So buckle up and we'll get started soon. That's about all the logistical things. For any of you who aren't familiar with the Tug Hill Commission, I think some of the attendees here are repeat offenders, but looking through the registration, there were people from all over the Northeast and some from all over the United States. So the New York State Tug Hill Commission, to introduce, is a small non-regulatory state agency charged with helping local governments and citizens shape the future of the Tug Hill region. One of our key areas of work is natural resources and the environment. And so our goal of this webinar series has been to enlighten and educate citizens of the Tug Hill and the region and afar about natural resources, environment, wildlife, etc. And hopefully you leave with some additional understanding and appreciation for those things. I'll go ahead and introduce our speakers for tonight. Our first speaker will be Paul Miller from Trout Unlimited. He is a retired Marine Corps officer. After leaving the Corps, he and his family returned to his family home on Fish Creek at the foot of the hill in the town of Vienna to carry on a 150-year family residency while serving Madison County. Paul is an avid but unskilled fly fisherman who advocates for native species as the Region 6 Vice President for New York State Council of Trout Unlimited. He is also an active member of the Tug Hill Black River chapter of Trout Unlimited and serves as an officer in Trout Power and the Fish Creek Atlantic Salmon Club. Paul served 19 years on the board of Tug Hill Tomorrow Land Trust. Second, we'll have Chris Murphy. He's a high school teacher in Vermont who is involved with Trout Power, a nonprofit organization that is working to identify and map the genetic diversity of brook trout in the Adirondack Park and surrounding areas in New York State. So thank you again to you both, and I will hand it over to Paul.
Thank you, Gabriel. I should have put in my bio that I'm a great fan of the Tug Hill Commission. I was introduced to the Tug Hill Commission my first month home from the Marine Corps. Went to a town of Vienna planning board meeting because I'd never been to a planning board, and there was an employee there from the commission that just captured my imagination at what they were doing. So over the last 30 some years, my admiration for the commission has not waned. Great organization, and it's really great to see the caliber of people that work there and the work that they do. But we came tonight to talk about fish, so I better get started. It's my pleasure to represent the New York State Council of Trout Unlimited and the local Tug Hill Black River chapter of TU. I belong to a class of sportsmen known for exaggeration. Yup, I'm a fisherman. Nearly all of what I'll talk about tonight is true, but just in case I lose my place and get creative, I've been advised to start with this ancient disclaimer. Lo, the angler, he rises in the morning and upsets the whole household. Mighty are his preparations. He goes forth with great hope in his heart, and when the day is far spent, he returns smelling of exertion and strong spirits, and the truth is not in him. Now that we've got that out of the way, let's move on. soon as I find the arrow. Not there.
Just give her a click, Paul.
Okay, next slide. There we go. In 1918, the Camden paper reported some big stories. Grace Peck and William Harrison Mills were married. Eva Stoll and Ray Stevenson got engaged. And there was a violent electrical storm that damaged the church in McConnellsville. 3 area barns were destroyed by lightning. It burned some hay and killed a hog and a pig. I don't know the difference between a hog and a pig. Didn't know there was one, but that's the way it was reported. And most of all, anglers were catching 3 to 6 pound brook trout from the Salmon River at Redfield. About the same time, a famous fly fisherman from Rome named William E. Scripture Jr. was telling about 3 to 5 pound brook trout being caught in Tug Hill's east branch of the Fish Creek. In Robert W. Streeter's book, The Greats of Adirondack Fly Fishing, it was emphatically stated that in Scripture's time, the East Branch was a widely renowned fishery. Scripture fished all over the country, but always said, like me, Fish Creek was his favorite, especially the area between Taberg and the Fish Creek Club, where Scripture was a founding member. And just a comment, isn't it interesting how Tug Hill often gets lumped in with the Adirondacks? There's always something that makes Tug Hill interesting and worthy of comment.
I think you jumped ahead there, Paul. 1 or 2.
I did. I did. I'm having trouble getting back.
Huh. Where's your little...
Gabe, can you get me back?
I can't control the slides.
Yeah. There you go, Paul. Go back.
No, I'm going forward.
Are the arrows on your keyboard working, or?
Oh,
Yeah, give that a
okay.
shot.
Let me try those.
There we go.
Yeah. It's a little no, that's inconvenient, but
Technology's great when it
there
works.
we go. There we go.
are.
So when you talk about the weight of a brook trout, the first thing is scientifically brook trout are not trout. They belong to the genus Salvelinus, and they're technically a char. Now, the biggest brookie I caught last year was probably 10 inches, and it would have weighed a mere 6 ounces, like this fish in the circle on the left that I caught and released, and he hung around long enough for me to get his picture. And the one being admired by my friend and local author, Mark Usick, is on the right. The 3 pound brook trout would be between 19 and 19 and a half inches. A 5 pounder between 22 and a half and 23, and a 6 pounder would be the fish of a lifetime, over 2 feet long. It's widely accepted that fish were bigger in the past, especially brook trout, and something happened in the last century that has impacted the ability of brook trout to reach their full potential all over their native range. This is what happened. We damaged their home. Both of the tails of big brook trout that I just told came from a time before dams, a lot of culverts, and other man-made obstructions to fish movement were heavily impacting fish reproduction. These structures prevent the fish from migrating to forage, to find mates, find spawning sites, and generally living as fish had evolved. Deforestation removes shade from stream banks, creating the conditions for warming water. Without living tree roots to hold the soil, after we cut it down, we had a lot of bank erosion. The banks caved in, the deep pools filled in, and the streams widened, become shallower and warmer. These shallower streams didn't clean themselves naturally of naturally occurring fine sediment. The stream bottom silted in, blinding the gravel beds and shutting off upwelling groundwater that brook trout need to spawn. There were fewer places for fish to hide in this degraded habitat. Fisheries declined. We took more out of our rivers and streams than Mother Nature could replace. When we fished, we killed the big fish, the 3 to 6 pound breeders that produced hundreds of eggs instead of a 10-inch fish that might produce a few tens of eggs. The practice of stocking non-native species like brook trout, like brown trout and rainbows, was intended to improve declining fisheries, but the non-native fish competed for habitat and food. They ate the young brook trout and the eggs. Brook trout that survived were pushed into subpar streams or into small headwater enclaves where growth and abundance are limited. Pollution happened. We used our streams as septic systems where waste depleted oxygen and further warmed our waters. And although acid rain has been declining, atmospheric deposition of pollutants contributed to the challenges brook trout faced, especially in the high elevations of the Adirondacks. And now climate change is a major limiting factor because of the profound impact it has on cold water habitat. Extreme flood events further damage stream bottoms and banks. Brook trout and other cold water species are in peril. Reversing this trend is something that Trout Unlimited and its nearly 400 local chapters have been battling since it was founded in 1959. All of these threats still exist, but we understand them better, so mitigating them when we can is the best thing we can do to conserve trout. TU made catch and release fishing popular, and today over 80% of trout anglers in New York practice catch and release. Catch and release is not the final solution to trout conservation, but it does help. Too far. So when we talk about cold water, what do we mean? This is a handy angler guide that conveys the idea that water, 70 degrees and lower, is a good description for cold water. Above that temperature, trout struggle to survive, and any brook trout caught and released at this temperature is at the risk of dying due to stress. Brook trout like their water cold. Below 65 degrees here is great, so you can catch and release fishing all day with little risk of harm. If the temperatures get above that, we suggest that you modify your approach by quickly landing and releasing the fish, or target other species, or better yet, just stop fishing and never fish above 70 degrees if you're going to practice catch and release. A little out of order here, so I'm going to go back up. So Trout Unlimited has emerged as a premier cold water conservation organization advocating for wild trout and salmon and their habitat. Our mission commits us to care for and recover streams and rivers so that our children can experience the joy of wild and native trout and salmon into the future. We know we can't do that on our own, so our vision includes working with stakeholders and communities to engage the work of repairing and renewing our cold water rivers and streams. There are 4 things that guide our practice. We need to protect wild trout and salmon in the places where they still exist. We need to reconnect waterways and the natural structures by removing obstacles that prevent movement and migration. We need to restore populations of native fish to places where it's possible and where they should be. And we need to sustain those cold water fisheries through advocacy and education. Some say TU is a conservation organization with a fishing problem because fishing is something we like to do in excess. That's probably why Trout Power and Trout Unlimited worked so well together, but we'll talk more about that later. Did it again. So let's define some terms that you'll hear when people talk about fish conservation. The words are wild, native, exotic, and invasive. I think we all have a pretty good idea of native, what that means. Native trout are a species that occur naturally in the body of water where they're found. They're indigenous. We can imagine that their heritage extends over millennia. In the context of this discussion, brook trout are New York's only native trout, and the true native strains have hung around since the last ice age. Wild trout, on the other hand, are those born in a body of water where they reside, but their heritage is not necessarily native. Wild fish are not necessarily indigenous to the region, but could be there through the efforts of man or some catastrophe, and they subsequently reproduce naturally. So a native trout can also be wild, but a wild trout is not always native. We sometimes refer to fish as exotic or invasive. Now I'll give you a quick identification lesson for those of you who don't fish. The two fish on the bottom are brown and rainbow trout. The brown and the rainbow have dark spots on a light skin. The top fish is a brook trout. Its markings are just the opposite, light markings on a dark skin. Other hints: the brook trout have the white leading edge on its fins. The browns have some red or orange spots, and rainbows, a pink hue on the gill plate and flank. In fisheries jargon, a species that is not native is referred to as exotic, regardless of how pretty or plain they may be to the eye. An exotic species that is harmful to the environment are called invasive. So browns and rainbows are exotic here in New York. Browns are native to Europe. Rainbows are native to Pacific drainages. Both were brought to New York to supplement declining fisheries under a somewhat faulty belief that they could replace our native brook trout. A fact that many find interesting is that brook trout are considered exotic and invasive in some places in the American West. They're not native there, but have introduced to Western waters through stocking programs initiated to mitigate the decline in fisheries due to dam building, loss of natives, or to diversify the fishing experience. In Yellowstone Park, brook trout are definitely considered invasive. Anglers must kill browns and brook trout in certain park waters in an effort to protect the native cutthroat trout, a special type of rainbow. I want to give a shout out here to Steve Hurst, the DEC's fisheries chief, and his numerous staff for their new trout stream management plan, which replaces a decades-old program, and it ushers in a new era of trout conservation in New York. The plan places more value on and better protects persistent populations of wild and native trout. The plan covers nearly all inland streams open to the public. It tries to balance DEC's mandate to provide quality angling opportunities to the public while preserving the state's aquatic heritage. It covers streams on public lands like state forest, the Adirondacks and the Catskills, and wildlife management areas. It also includes waters where DEC has obtained formal public fishing rights from landowners. Under the plan, DEC will no longer stock fish where self-sustaining wild populations of trout exist, be they brook, brown, or rainbow trout. Also, the DEC will not stock fish on land where there is no public right to fish. Currently, DEC is working on a new plan to manage brook trout in Adirondack ponds and lakes. It's a little bit different challenge, but in both plans, native fish are given priority over exotics and wild fish given priority over stocked fish, with the highest priority given to brook trout where they can self-sustain a wild population. Too far, Sue. The matrix on this slide demonstrates a hierarchy of stream management and classification based on several factors. The first is the stream's ability to maintain self-sustaining populations of trout. If it does, a stream will fall into one of the three categories on the left. Wild means it just supports trout reproduction. Wild quality and wild premier designations indicate progressively bigger and more productive streams where reproduction exists. If a stream doesn't support a self-sustaining population of trout, but will hold trout, then it may be categorized as stocked, like the column on the right. Analogous criteria for size and angler opportunity differentiate the stock stream categories. Another aspect of the new management plan is the high priority given to the habitat for fish. It's a commitment on the part of the Department of Environmental Conservation to the idea that if we build it, they will come. New York DEC, Trout Unlimited, U.S. Fish and Wildlife, Department of Agriculture, and other resource agencies are all joining in to fund stream restoration, removal of obsolete dams, and the reduction of culverts and other obstacles that impede fish movement. Added to the objective of these stream restoration projects is the imperative to connect streams and rivers to their floodplain in order to restore natural stream function and to minimize the effects of rain and runoff events made more destructive by climate change. I have a heavy finger here tonight. It's keep going too far. About the same time that DEC was updating its management plan, Trout Unlimited's national organization was working on a strategy to guide its chapters and staff in the work that they do. It was an effort to identify the right places to invest money and efforts to protect trout and salmon in their habitat. What resulted is called priority waters. Using known data on habitat, fish populations, land ownership, ecology, and a lot of input from local people who fish, a map of priority waters was developed for every state. The effort took into account strongholds of cold water habitat, existing climate resilient and genetically diverse populations of fish, and places where management principles could be applied. Stream production and restoration in these areas will receive TU's strongest support over the next 5 years. Priority water designations will be refined as new data becomes available or as work helps to improve stream conditions. The program goals align exactly with DEC's new trout stream management plan stream categories. And here's the map of the priority waters in New York State. That northernmost region up here, this little, now it's doing that. Oh no. Going back. Can't go back. I apologize. I'm having a real problem here. There we go back.
There you go, Paul.
Yeah.
Good.
So this is the map of New York's priority waters. And although this area, this large area here 4 is called Adirondacks, it does include Tug Hill, this lobe over here that I'm pointing out. Want to switch gears here now to showcase some of Trout Unlimited's on-the-ground projects that are making a difference in the designated priority waters closest to home. This work is facilitated by a group of Trout Unlimited employees called the Northeast Cold Water Habitat Team. I won't say as much as I could about this group, but I don't think I've got enough time here to say enough. They do really good work by assessing stream conditions and identify projects that would improve habitat for fish. They design the projects, undertake permitting, management, and construction of stream restoration projects. The current efforts are focused on projects that involve faulty culverts and other barriers to fish movement. Making culverts passable for fish is a low-cost but highly effective way to improve fish habitat. Other benefits from these right-sized culverts are to enhance stream function, flood resilience, and they decrease road and culvert maintenance. Notice the culvert in this photo on the left. A fish can't negotiate upstream passage through that culvert because of the height of the drop of water coming out of it. By comparison, look at the structure on the right with plenty of water under it. A fish would have no trouble going upstream or downstream, wherever he wanted to go. That's a condition that reconnection projects seek to create through fish passage design and placement. Here's a watershed map of the Moose River, a tributary, we know, to the Black River that's a known stronghold for native trout. The cold-water habitat is engaged in the study of this watershed to identify and categorize every barrier to fish movement. They're depicted here with little red dots. There's dots of various colors. There's over 80 of them. Some of the work is done in the office by computer, but ground truthing conditions takes days and weeks of field work. And in the end, the goal is to identify reconnection improvements that get the most bang for the buck. Remember the mantra I mentioned earlier? Protect, reconnect, sustain, restore, and sustain. These culverts are a big part of the reconnection. I'm going to look at a couple of projects in the Beaver River watershed that the Coldwater Habitat team identified through a study that they did of the Beaver River watershed using mitigation funds from the Beaver River hydro projects. So this map shows a location of a culvert in the town of New Bremen, where the red dot is, was identified as a fish barrier. Replacement of that cover with a fish-friendly structure reconnects over 3 miles of quality wild trout stream. The U.S. Fish and Wildlife Service, Trout Unlimited, Lewis County Soil and Water Conservation District, and the town of New Bremen have all come together and collaborated to come up with a design of a replacement structure. They found funding, obtained permits, and contractors will replace this culvert, which is scheduled to happen in the summer of 2024. Here's another Beaver River watershed project in the village of Krogren on Warner Creek at Long Pond Road. This project has the same partners, except a different town is involved, and it reconnects over 10 miles of trout habitat, some 4.9 miles upstream and 5.8 miles downstream, leading to the Beaver River, which is even longer. Now, it's dangerous to anthropomorphize wildlife, but I'm going to do that. Let's say you live in a 1,000 square foot house on 1 acre of land, and all of a sudden you have a 2,000 square foot house with 2 acres of land and access to a new public park down the street. Wouldn't you feel like your life was looking up? If a fish can feel joy, these projects will make them pretty happy. Most practically, it's common observation that projects like this see immediate use of the new structures by native fish. As time proceeds, measurable increases in migration and reproduction are observed. So I'm going to take a break here, and I'm going to turn this over to Chris. Chris, I hope I can do better on the slides with you than I did with myself. I'll come back in and talk a little bit toward the end.
Thanks, Paul. Appreciate it. Hey, everyone. So my name's Chris Murphy. I am the current president of Trout Power, and Paul is part of our board. He's our treasurer and kind of one of my right-hand guys. And so what we're going to talk about is that there's been a collaboration that's happened between the Tug Hill Black River chapter of Trout Unlimited and Trout Power. I, to be transparent, personally did not have a tremendous amount of involvement. Paul was our real bridge between TU and Trout Power. So what my part is going to be is talk a little bit about first off, what Trout Power is as a nonprofit and what we're doing. And then Paul will speak specifically to what was done up on Tug Hill and what some of the results were from that mission. So in terms of what Trout Power is, again, as mentioned, we're a 501c3 nonprofit organization, and our overall mission or mission statement is to enlist the power of anglers to protect, restore, and enhance native brook trout populations and their habitats across their native range through citizen science, advocacy, and stewardship. Now, in terms of what that actually means, because the mission statements are great. And then you're like, okay, so what do you actually do? So first and foremost, we are an organization that relies on volunteers. And specifically, we're taking volunteer anglers, fishermen, fisherwomen, and turning them into citizen scientists, which I'll talk about in a minute. Through those volunteer efforts, we have our own ongoing solo projects that we do. We've also worked with groups and organizations, as we're talking about tonight, with groups like Trout Unlimited, DEC, and others. And then part of our citizen science piece is that while we're doing a lot of the actual field work, we work with the universities on projects, including the genetic analysis. That's kind of the back end of what Trout Power actually does. So citizen science, this is something that I think is one of the most unique and one of the coolest things about Trout Power and our group. And I say that both as a member of Trout Power, as a fisherman, as a high school science teacher. Citizen science is a really powerful thing in our world these days, not just for trout. But citizen science is using volunteers, amateur volunteers, to do part of or all of a scientific process. So for us at Trout Power, we're using volunteers to actually go and do the fishing. And I'll talk about what we do when we actually catch a fish. And then we turn it over to the pros at universities to do the genetic work and the analysis for us. So it's a really cool way to get people involved in that. Paul, can you go back one slide, please?
I'll try.
Okay. That one, perfect. So when we talk about citizen science, taking volunteers and turning them into scientists, if only for a weekend, there's a lot of really great things about citizen science. First off, and this is one of the reasons that we've been called on a few times by the DEC to be involved with projects that they're working on, is that it's economical because all of us are volunteers. And not only volunteers, but passionate volunteers. We had our big event last year where we had 24 volunteers show up from 7 different states. And we have another sampling event coming up next month where we're up to 30 volunteers from 8 states and 2 different countries. Our neighbors to the north, we've got a few people coming down. But people get really excited about this. They volunteer their time. And because we get a lot of volunteers, that allows us to cover a lot of ground. So when we go out on a weekend for Trout Power, we can get out there and collect a lot of data. We can catch a lot of fish. Thankfully, a lot of our volunteers are really great fishermen, which helps people like me and Paul who are, we're good. We're good enough, I guess. But it gives us the ability to catch a lot of fish, gather a lot of data, and cover a wide, expansive area. And then the other things that's great about citizen scientists is that it's a way to educate and connect the public like we're doing here tonight, which is a lot of fun for us, and give them a specific cause. Whether it's the next time you're driving, over a culvert, maybe you look at it a little bit differently. Whether it's looking at a stream that's baking in the hot summer sun that's a lot lower than you normally see it. This is a really great way to do outreach for all of these different causes, not just brook trout conservation. And connecting it and outreaching to the public. And last, it's fun. We have a Trout Power, folks. We have a lot of fun, and it's quite a good time. Go ahead, Paul. So in terms of Trout Power and what we do and how we do it specifically, the biggest thing that our organization is involved in is doing genetic analysis of brook trout. Since 2016, our group has really focused on looking for and at the genetic diversity of populations of brook trout in the Adirondack Park. Genetic diversity is something that, whether or not we recognize it consciously, it's a really important thing for all species. And species that we have on this earth, including ourselves, that are able to reside through different changes in climate or habitat. Genetic diversity is what really allows that adaptation and pushes that adaptation, which can lead to survival for example, brook trout in the Adirondacks. If you've never really been able to recognize the genetic diversity, that's understandable because it's hard to see. I think the best way that you can view it is if you are someone who likes to catch fish and enjoys fishing. Next time that you catch a fish or you catch a trout, especially if it's a brook trout in the Tug Hill region or the Adirondack Park, Take a look at it. And then if you go catch another one somewhere else, there's a pretty good chance that they may have subtle variations. If you look at the two fish that are on your screen right now, these are two fish that are from two different watersheds, not separated by a tremendous difference in terms of geography, but from different watersheds in different areas. If you look at the fish that's over on the bottom left, and you look at the side and the flank, it's a very dark fish. You actually can even see some of that cobalt and that purple and violet down kind of on the side flank and down on the belly. And then looking at the fish on the right, vibrant yellow, the vermiculations, those kind of twists and turns darting along its back. Just, again, the same species of fish, both from the Adirondacks, but you see that different expression, and that's kind of a sign. It's not enough to say that they're genetically different from each other, but it's one of the things that we can see and say, hey, isn't that neat and isn't that beautiful? We're lucky that our fish that we catch are pretty fish. Go ahead, Paul. So what we do when we catch one of these pretty fish, that's why I complimented them, call them pretty and beautiful, is that when we catch these fish, we take a tissue sample from them. Now, as sometimes this image looks a bit dramatic, especially with the flames over a set of blades. But what we do with these fish is that, well, first off, we handle them really carefully. What we very often do is when we catch these fish, we'll put them in a Ziploc bag that's filled up with water right from that stream. One thing that's really important, if you are someone who practices catch and release when you catch fish, is keeping those fish wet and keeping them in the water as much as you possibly can. I'm the first one to understand taking a fish out of the water for a moment to look at it, to look at its colors, to appreciate what you've just been able to experience and that gift of nature. But one trick that I always say to people is that, think about a fish, right, being out of water. It's kind of like you being underwater. So if you take a fish out of water and if you hold your breath, right, as soon as you start to struggle because you're not breathing, well, that fish was struggling too, especially with trout. They're really fragile fish. So we put them in bags so they're able to sit and take oxygen out of the water and be nice and happy. It's a great chance to take a look at them. While they're safely in the bag, what we do is we take a pair of small scissors and we sterilize them using a lighter, butane lighter. We burn the scissors on both sides. And then really carefully, we take a really small tissue sample from the top part of the tail. Once we take that sample, we put it into a vial that's full of ethanol. And then as soon as we have that sample in ethanol, our first and foremost priority is getting the fish back into the water. So we are completely catch and release. Every fish that we sample immediately goes back to exactly where it was caught. In terms of the location that it was caught, we mark this using GPS, whether it's using a logging app or using it's an actual GPS itself. We take the coordinates of where that fish was caught, where the tissue sample was taken, and then we re-sterilize the scissors. So we're making sure that we're sterilizing those twice in between every time that we take a tissue sample from a fish. Go ahead, Paul. So that's a great drawing that was done by one of our members. This is actually what it looks like a little bit more in the field. So catching a fish, there's an example of a great fish in a bag. Look at that. So that's actually self a shout out here. That's my dad with a big smile on his face. Then next we, really carefully take a fin clip sample from the tail. You can see there's my dad again, and our volunteer Ryan, and our founder and current member JP Ross holding and standing guard, watching close by. In the third segment, you can see, that really small tissue sample being taken out. We've got all of our vials, our little microcentrifuge tubes down in the bottom left. We map the location of the fish, and then, as you can see in the bottom right, we send it on its merry way to live a full, healthy life, maybe a bit traumatized by the out-of-body experience they had, but again, we do this really carefully, and we do it really quickly and get those fish back in the water. So I'm pretty confident that they're doing all right. So how we do this in terms of our actual tools is that our volunteers receive what we call a fin clip kit. You can see kind of a picture in a plastic bag so you can see the contents. They've got these microcentrifuge tubes, again, with a diluted ethanol, a pair of scissors, a lighter to sterilize those scissors. Our volunteers get a log sheet so they can log the data of where these fish are being caught, what the vial number is, and again, where exactly that fish was released. And then again, we send people out. We're not sending them out blindly into the field. We tell them, hey, we'd like you to go to this stream and fish from here to here and see what you can catch. And we train them. We make sure they're good to go. And then we send them out there to have a great time, do some citizen science. So when we look at the type of genetic analysis that Trout Power did between 2016 to 2019, it's called microsat analysis. The reason I bring that up now, even though it's 2024, is that this is the kind of analysis that was done during the collaboration period between Trout Unlimited and Trout Power. So what I'm going to do, I'm going to go into science teacher mode. Appropriate enough. I'm in my classroom right now, so I'm going to go science teacher on you for like 3 minutes. So bear with me, and I'm going to explain how this works using a few images. So we have 3 fish, and as you can tell, they're clearly the same image, just hooray for color filters. But let's say that these are 3 separate fish and 3 different populations. So top fish is population 1. Second fish down is a hybrid, so meaning it has genetics of population 1, and it also has some genetics from a hatchery population. So that fish, the third fish that's a little grayscaled, there's an example of fish that may have been or probably was stocked by the DC or maybe a private stocking. And what we've seen and what we have data to show is that if you put stocked fish in areas where wild fish reside, that they can breed with each other. If you know anything about how a fish reproduces, it's a pretty inaccurate process in terms of aim. They dig nests, the females lay eggs, the males apply to the eggs, and so there's not a lot of rhyme or reason other than, hey, you're the same species I am, so we can reproduce together. So we've seen examples where we have wild fish that are reproducing with stocked fish. The issue there is that these wild fish have adapted over hundreds, if not thousands of years to that specific habitat. Meanwhile, those hatcheries fish aren't. They were raised in, let's be frank, a concrete tank, and stock fish are great, and they're great to have in certain areas, but not where wild fish are already surviving and if not thriving. So as Paul mentioned, this is why we applaud Steve Hurst and his DC team for their stream management plan and saying, hey, if we have fish like you have in population 1, where they're wild, where they're surviving, where they're building and keeping their population strong, then we don't need to interrupt. We don't need to go in and throw a bunch of other fish in there. We just need to make sure their habitat is good and that they can keep on surviving. But I digress. When we look here, the reason I bring this up is that when we take our fin clips and we send them to our university labs, they extract the DNA, they sequence it, and then they analyze it. They do the real sciencey stuff. And then what we get back is that we get back bar plots or bar graphs, if you will. So if you can look at population 1 and then our second population and our third population, over in the bottom right corner, you can see examples of these bar plots and what they look like. One vertical bar represents one individual fish. And so this is the way that we get our information back and then can do a comparative analysis of what the genetics actually were for those fish. So Paul, if you don't mind going to the next slide, please. What we compare to is that there are 6 strains of brook trout that the New York State DC stocks within the Adirondack Park. Those are the 6 that are listed up at the top of your screen, pardon me. So the domestic fish, the Temiskaming fish, the domestic Temiskaming hybrids, that are primarily put in catch and release areas. And then the 3 on the right, the windfall, the little tupper, and the horn strains, those are strains that are actually strains that are native to the Adirondacks. For example, the Little Tupper Lake strain. And those strains are then taken by the DEC and then populated primarily in brook trout ponds. So if we look down on the bottom, what we see is that we see samples that were taken by Trout Power and were taken over years and taken in these various areas. So what we do is that we do basically a visual comparative analysis via color. So if you look up on top It's kind of like that childhood song, you know, like one of these things is not like the other. And I promise I won't sing anymore. I have terrible voice. My apologies. But if you look up top, you see these colors. And then when you look down on the bottom, we're basically just comparing the colors. So if you see a color down on the bottom, whether it's the yellow in the bottom left or the pink in the bottom right, those are showing unique populations or unique genetics, pardon me, in those populations that don't show up when we look at the stock strains. So Paul, if you don't mind going to the next one, please. Here's just one more kind of reinforcement here. This is good teaching practice. Show something and then show it one more time differently to make sure you got it. So over on the left, you see the stock strains. And then here's an example of some samples that Trout Power volunteers collected in 2016. So again, when you see that blue coloration that you don't see over on the left, that's showing that those fish have unique genetic markers that we don't find in the strains that the DEC are putting into our waters. Now, certainly recognize that you do see some of those colors, especially within this population. You see some of that Temiskaming hybrid coloration showing up. And again, there's that hybridization that we mentioned before. So evidence that somehow, some way, most likely from stocking in a lake, you know, these fish were hybridizing a little bit with some of the fish that were stocked in a nearby lake. Awesome. Go ahead, Paul. So that's my lesson here. So now what Paul is going to do is he's going to take back over the reins and talk about when our Trout Power, you know, some of our volunteers, thanks to Paul, linked up with some Trout Unlimited volunteers using that protocol that I described. These were what we found in the Tug Hill area and what the results were. And hopefully if I was decent enough at teaching, you'll be able to recognize these colors and these bar plots. So take it away, Paul.
Thank you. So on the left-hand side of this display, you see all of those stock strains shown again. The colors may be a little different. The color's not really important. What's really important is the differences. From this line over, can you see, am you seeing my cursor, Chris?
Yes.
Yeah. From this line over are all of the fish that we caught had analyzed in our Tug Hill study. There were 33 fish. The deep purple color is not like any of this. And the purple is really deep and there's almost nothing else, almost no other colors on each sample. So this was one stream from here to here. This was another stream. There was another stream here. And oh, wait a minute, what's this blue stuff and this green? Well, this blue is the same as the Temiskaming or the domestic Temiskaming hybrid. The green is the same kind of as the little tupper. So we've got some fish there that looks like their genes have been crossed with somebody else. How come they don't show over here? The reason they don't show over here is because there are 3 fish here represented by these samples with the odd colors. Those 3 fish were caught below an obstacle that is too big for them to pass. So in the water downstream where where some of these hatchery fish were stocked, they're not able to get into the headwaters where these Tug Hill fish were. This is an incredibly pure population of fish. We can definitely say those are native. There are probably a lot of you on the call on the thing tonight that know there are native fish on Tug Hill, but boy, here is the scientific proof that they're there. So this was really astounding to us and really gratifying when we got these results back. Pretty strong and pretty profound. So going back to this picture here. So what good is citizen science? When Trout Unlimited started putting this map together, it was pretty much of a no-brainer that the Adirondacks and the Catskills were going to be in these headwater or in these priority water areas. It wasn't so obvious that Tug Hill was going to be there until this study got out. People took notice, and Tug Hill became a no-brainer for Trout Unlimited to put it in its priority waters program, where going forward, any stream projects that we identify in the Tug Hill area will receive priority for the cold water habitat restoration team to do their work and for us to seek funding for those projects. So pretty neat work and really gratifying for those of us. And the team that went out and did that, I'd say we're half and half, Chris, when you get right down to it. Probably half TU members and half Tug Hill TU and half Trout Power people.
Yep.
So we're getting close to the end here. Trout Power has a really robust social media presence. All you got to do is look for Trout Power and you'll find them. If you want to know more, there's a lot there. And Trout Unlimited has a really strong internet presence at tu.org. So you can find out a lot more there and learn more than you ever wanted to know about fish. So I'm done. If I talk anymore, I'm getting to the point where I might start exaggerating. So here we go. We got quite a few in the chat. Gabe, I don't know if you've been able to look at those. I have not.
Yeah, we have a couple questions.
Yeah.
First one is, are there ways for folks in our region to get involved with the Black River chapter of Trout Unlimited and/or Trout Power, and how would they go about doing so?
So if you go to the both organizations have Facebook presence. You could search them out on Facebook and find us, and that's generally where we publicize things that are coming up and what we're doing. The Trout Unlimited chapter meets the second Tuesday of every month in a cafe, bar and grill in Pulaski, and it's called 11 North. The best way to find out what's going on with Trout Power is to become a member. When we plan our outings for Trout Power, the members get priority to get a slot to go. You know, you're taking 30 people out into the woods where a lot of people don't go. So we need to control the number of people and how they're going. So you need to be a member of Trout Power to do that. On the Trout Unlimited website, there's a link that says find your chapter, and that will put you in touch with us as well. Oh, and also you can find us on Facebook. Next one. I hope I've answered that well enough.
Yep.
Yep.
Next one is, what did Trout Power, Black River chapter of Trout Power, Trout Unlimited and Trout Power look for from a partner organization such as Tug Hill Tomorrow Land Trust?
If you have, everybody has their own special water. And if you have a place that that you think is special and you'd like us to try to work it into our plan to sample, get in touch with us and we'll try to develop a plan. We're planning now through this year in both organizations, and I think so we'd be looking at least next year and maybe longer in our planning horizon. But we're looking for people with interest, people who have water that they think is unique, and they want to know more about it. When Trout Unlimited gets to their projects with the Northeast Cold Water Habitat Team, they're unique in that they are only partially funded, and most of their field work is funded at a project level. So they have to find a source of funding before they can go too far with it. And they've been fortunate with some of the programs that are out there by the U.S. Fish and Wildlife Service, by DEC, and as I mentioned earlier, the hydropower mitigation funds that are available in some places. So funding is, like everything, time and money. Never have enough of either, but you need them both in order to get these things done. Yep.
I have a question myself about that last, and then I'll get to another one in the chat. About the last chart showing the genetics of the Tug Hill trout. You said that there were 3 in there that had non-native genetics, and those came from below a barrier. Was that barrier natural or man-made? And if it was man-made, would that incentivize leaving that barrier to continue to leave the pure native trout isolated?
It's a knife that cuts both ways, isn't it? And so it would depend. It would depend on what you find in the area. You know, if there are trout there, it's probably a cold water stream. At some point, there may be, it may meet up with warm water, warmer water. If there are a lot of warm water species that are in the areas downstream, then, that might not be a good plan. But, in the long run, we're trying to, Trout Unlimited is trying to affect the water quality and the habitat quality in a watershed, not just in a single stream. So you have to look at the whole picture. Yeah, it can go both ways. That's always something that people bring up, and you got to work with it and solve the problem. Hope that answers.
Yeah,
like most things,
great.
like a lot of things, it depends,
Yeah. I have one comment, one last question, and then if there are no more, we'll wrap up at around an hour. Jessie Vidala is from Trout Unlimited's Northeast Restoration Team. She said that she personally would love to connect with more local land trusts within the region to help support our Trout Unlimited's shared goals in cold water conservation, and her email is in the chat if anyone would like to reach out. And another question.
Let me make a comment.
Yep.
Jesse's a he.
Oh, sorry about that.
And Jesse is the outreach coordinator for the Northeast Cold Water Habitat Team. He was just at a meeting last night and talked about those very same projects in the Beaver River that I talked about in my presentation. He provided me with those slides. So the team is headed up by a woman named Tracy Brown. I think they've got about 8 people that are on their team this year. It's a mixture of some permanent and some seasonal employees. They really got an appreciation for the Adirondacks when they went into the Moose River Plains area. Lot of real backwoods there. That's an area that Trout Power has been as well. So it's an area that's definitely getting a look because it's a stronghold for cold water and for some unique populations of brook trout. So Jesse's a great guy to get in touch with. That whole team is really strong. A lot of the time there's nobody home. They're out of the office. Places where they typically go, there may not be cell service, but they are talented, and they'll teach you a lot. There's a lot that people can do, and Jesse's the guy to talk to if you want to get personally involved in this sort of stuff. A lot of times their projects involve contractors, heavy equipment, and they don't want a lot of people mogging around out there to get in the way and potentially get hurt. But when that work is all done, there's always a day or two at the end of a project where they do tree planting and some other things. And it's a good opportunity for individuals or groups to get involved with this stream restoration. Okay, go ahead. Next question.
Thank you. Yeah, we have a couple more questions and a couple raised hands, so we'll try to get to as many as we can. If cold water species like brook trout are negatively affected by increasing water temps, does that mean that fish more tolerant of warmer temps are doing well? Or is the change negative across the board for all species?
I think I'd have to say that warm water fish are doing better.
Yeah, I would say
think
well, it's
we found smallmouth bass, which are kind of a crossover between cold water and warm water, but we see smallmouth, moving into a lot of places, largemouth expanding. Some of the other warm water species are expanding their range, and it's a real threat to our cold water species. So I think that really points out how important it is to protect them where they are and to protect their habitat, because the world's closing in on them.
Great, thank you. Someone else said leave a message at the Tug Hill Black River Trout Unlimited 589 Facebook page and they'll get back to you. Another question, is there a list of streams that have already been sampled on Tug Hill?
There is. It's in my back pocket and you're not going to see it.
A true fisherman. Nice.
And we do that for a reason. We don't want to put a target on our streams. I will tell you that all of the streams that we looked at on Tug Hill are public fishing rights. So you have a right to go there. You can find it yourself by going to the DEC website. And if the further upstream, the further you get away from people, I think the more likely that you're going to find exactly what we found. I think everybody involved in Trout Power is pretty flabbergasted that we finding these native fish a lot more than we really ever thought we would.
Yes. Great. Well said.
Another question, how do we tell a stocked fish from a wild one?
Question.
Chris?
Sure. Stockfish versus wild fish. I think that most of it's going to be visual. If you actually, first off, if you catch the fish, and this isn't a great thing, but one thing that I notice overall is that wild fish usually fight a little bit harder. They usually bat a little bit more. Stockfish, sometimes I've heard the analogy a lot about the wet paper towel on the end of your line, but also I've caught fish that I know are stocked that took me for a ride. So that's not always the best way. I think the best way is probably visually. That stocked fish will, if you look at their tail, and if you look at the fins on the bottom of their body, very often those will be a little bit more worn down. Very often you'll see the tail edges a little bit rounded, and that has to do from the fact that as they were growing up within these tanks, these tanks very often are literally concrete tanks. So their fins and their tails are scraping up against this or other fish as they're growing. And that's usually the biggest thing that I see, is that if you catch a fish with full, vibrant, colorful fins on the bottom and on the tail, chances are that it's most likely a wild fish. And the only other thing too that I would suggest is that the DC does a really nice job of telling where they stock fish. So if you go and you fish a stream and you catch fish there and you want to know, hey, I wonder if I caught a lot of fish that were the same species and they were about the same size, you can go and check the stocking maps and check the stocking reports, and the DC reports all of the fish that they do stock. So if you go and look at that and you were fishing a stream or a river or a pond or a lake that isn't on that list, then there's a pretty good chance that you were catching a wild fish, which is really cool.
Yeah, I would say that location is a pretty strong indicator of what you've got. If you're fishing, below a culvert or below a bridge next to a road, and you catch 5 trout in there right away,
All 8, 10, 10 inches.
Good chance that they're
Yeah.
stock fish.
Yeah.
Stock fish are generally not quite as colorful. It's really profound in the fall when the brook trout, especially the males, color up. They get a brilliant orange color on their belly and on their body in places, and they look really something. Color is muted colors, probably a stalker. So that's about the best you can do. If you open one up and they're nice and orange colored, probably caught a fish that's a native. If it's kind of pale, it's probably a stalker. But then it's too late, So yeah. Next question.
I think that's about all the questions that we have in the chat. Is there any last comments that Chris or Paul you'd like to add?
I mean, I will add one. Paul, I'm gonna shout you out here. Someone, I think it was Jesse, called you a hero in the chat. And hero is a word that is used way too much, but it should be applied to Paul, because Paul doesn't talk about how much he's done for brook trout, for conservation in New York State, how much he's done for Trout Power, for TU. Paul is a pillar of trout conservation in the state. And yeah, sometimes his computer skills are, they're not an A+, but hey, we can't all be great at everything, right, Paul? So,
Yeah.
Paul, you are an amazing person, and you should be recognized, because you're hanging up there on Tug Hill, but what you've done for the state and brook trout in that area it should be commended. So
Okay, that's enough.
No, I get to.
That's enough of that.
I know. I saw the hero thing. I was like, you know what? That's a good use of that term.
Jesse and I do have a bromance going on.
Okay. Well, then you're cheating.
As I do with everybody on that cold water habitat restoration team. They're the best. They're the best thing that's happened to Trout Unlimited in New York State in the 35 years that I've been a member here.
Cool.
Without a question. They are the best thing that's happened to trout in New York State.
You call them heroes, right? There you go. Awesome. Yeah, but thank you, everyone. Thank you for listening. Thanks for following along. And fishing season's just begun, so get out there and go catch some fish. Have a great time. It's what it's all about.
Yeah, and thank you again to Chris and Paul, both of you. You are excellent speakers tonight. That was a lot of information. You got a lot of kudos in the chat, both of you. So thank you for being here tonight. And for everyone else that's in attendance, like I said at the beginning, you will send a follow-up email and you get a recording of this. If anyone is interested, point them to our YouTube channel. And we'll also send along the slides and any relevant links that were in the chat tonight. So thank you again, and I hope everyone has a wonderful night, and especially if anyone attended all 4 of the webinar series, I commend you, and we're on to spring. So have a good night, everyone.
Thanks, Gabriel.
Thanks, Gabriel. Thanks, everyone.